Yes. Okay. Now everything is get fine. So first of all, good evening to all. This is not a right time to take the lectures, but as on the part one, as on the virtual, we can take the lecture at any time. So last time we were discussing about the certain aspects of the types of the fermenters. Okay, so what is a fermenter basically? You know, there are the different four types only. One that we call them as in the a batch fermentation. Second, we call them as in the continuous fermentation. The third one we call them as in the fed batch fermentation, and the fourth one, this is we call them as in the solid state of fermenter. And last time we are studied the certain aspects of the submerge, different types of the submerged type of the fermenter, and one of that we are discussing regarding with the stir tank fermenter, another in the form of the what you call them as the bubble gas fermenter, and this is the another type of the submerged type of the fermenter. The name itself indicates one: air, lift, and fermenter. So air is responsible for this fermentation process. The airlift a bioreactor, which is just a 99%, which is similar with the bubble column a reactor one, but it is get differ by the certain important fact that they contain the draft tube, which is not been present in case of the what you call them as a bubble gas fermenter or the bubble column fermenter. Okay, so this is an additional function which is get present in the airlift fermenter. So what is the basically the use of the draft tube? The draft tube may be sometimes that will be the inner tube. We call them as in the airlift a bioreactor or the fermenter, which we known as in the internal loop. Okay, or most of the time they can be present as in the external tube. We call them as in the airlift a bioreactor with the external loop. So what is the use of this external as well as the internal loop? This loop basically which can improve the circulation of the oxygen, and they are get properly transferred. At the same time, they are equalize the shear forces which is get present inside the reactor. There are the large number of the forces which is get applied one whenever there will be the fermentation, either in the form of the stirring with the help of the agitation speed is there. Second one in the form of the oxygen transport, which is very important whenever it will be the aerobic type of the fermentation. I'm not talking about the anaerobic one. What's happened whenever there will be the anaerobic conditions are there. But most of the time, the industrial fermentation is totally depending upon the aeration mechanism is there. So we call them as in the KLA fermenter. How the oxygen it can get a transfer to the mass one. So this is an additional task is there. Now you have to know how this particularly the fermenters one, which play the important role in every task one for the XYZ manufacturing purpose one. So it may be the anything. Okay, now this is the basic diagram. The air lift a fermenter with the internal draft tube. At the same time, another on your particularly the left hand side as well as the right hand side. So one tube we call them as in the internal. Okay, so that inside the bioreactor there is a presence of the loop is there. So this is an internal loop. Okay, so this is an internal loop. So this is known as the external loop. So both this purpose, just like the bubble column, a bioreactor, in the same fashion, At the bottom, always there will be the presence of the asparger. So there are the different types of the spargers are there. What is the role of the sparger? You know very well. You have to introduce the oxygen or the air. It is always be a sterilized. Okay. And it is always from the bottom. We know very well what is the advantages from the bottom, because already the gases are which can get moved toward the upward direction. This is very simple task one. In the same fashion, when you are introducing the air, that is a sterilized one, 100% they can get mixed with all the essential, that will be the microorganism as well as the what you call them as in the media, which containing the large amount of the carbon, nitrogen, minerals, vitamins, etc. Along with that, it can maintain the proper temperature as well as the pH one. So how this particularly the mechanism, the air lift. So air is responsible for the proper transfer of the oxygen towards the microorganism. Whenever we are pushing the air from the bottom of the bioreactor, definitely there is a formation of the bubble, and this bubble basically they are get enters through the internal tube is there. So always the gas which can get move from the bottom toward the top side is there. Okay, and in case of the external one, so see here from this asparger, the air is get a push inside the fermenter. This is absolutely the sterilized one. Now when the air, it can get breakdown one in the form of the aerosol, that is on the bubble formation is there. So these bubble, they are get a move in a upward direction. So at the same time, so this can get move in this direction, go toward the upward side, and again from the upward side it comes back with the same mechanism. So always there will be the pushing of the air. So media as well as the oxygen, they are get a properly get a mixed one. That's why it is known as the air lift a fermenter with the internal loop. Now another part. So this is the an external tube is there. We call them as an the airlift fermenter. Okay, and that airlift fermenter who having the external loop, just like in a previous diagram. So always the air, sterilized air or the oxygen which is required for the microorganism, you have to push from the bottom with the help of the compressed air. Now always this oxygen which can get moved towards the upward direction in the form of the bubbles, and later on this media come out through one of the end. Okay, and it again break down in this fashion and it can get a circulate one. So this is known as the external loop, airlift a fermenter with an external loop. Okay, so are you getting this point?
Yes.
Yes or no? Are you getting or not?
Yes, sir.
Okay. Okay. Now, in case of the internal loop design, why this is required? Because every time we cannot use the batch fermenter. You can use the another type of the designing fermenters are there, and this design, the aerated arises as well as that things basically, sorry, regarding with the unaerated down corners are generally containing in a small shells. In case of the external loop configuration, this is in designing. When you are designing the fermenter, all things you have to keep in a mind. Which type of the fermenter you required whenever you are interested in a different types of the product. Either it may be the primary metabolites or either it may be the secondary metabolites one, either in the form of the carbohydrate, in the form of the protein, in the form of the lipids one. But what about those particularly the product which is acting as in the secondary metabolites and their production is very, very less one. And whenever the production is get less one, naturally the price of that product will be the high. And that's why everyone, that will be the pharmaceutical industries, they are manufacturing the near about till and date, near about 800 different types of the antibiotics, more than 20 types of the steroids. Apart from that, more than 50 types of the acids. And nowadays, all these industrial microbiologists, sometimes they are referred as the mine microbiologist one. They are using our microorganism for extraction as well as the purification of the metal ores which are present in the earth crust. So this will be the one of the emerging science where our microorganism basically we are utilized for the extraction purposes one. Now rises there down same one. Sometimes there are get a link near to the top one, just like in case of the previous diagram. So your external, particularly the loop, it is sometimes they are get linked towards the top as well as near to the bottom, and down the what you call them as in the commerce, that will be the downward flow is there, and the working aspect ratio of the airlift fermenter is 6 times greater than the batch fermentation is there. Now this is the another way that is the bubble column fermenter. Last time we are discussing this one. Okay, so right now we are not discussing this bubble column fermenter, but instead of that, we will discuss the another aspects of the another type of the fermentation process one. So which of these fermentation process? Last time we studied the continuous, and I'm saying I will show you the certain continuous fermenter tanks one. So in March, the 3rd March, we are visiting to the Sula Wines, which is situated in the Nashik, as in the industrial tour. And we are visiting to this greatest fermenters one. In this Sula Wines, near about a 35 huge fermenter which is get associated. And you see this. Also that will be the Sula Road, which is situated in the Savarga, near to the Nasik, near about 35 kilometers away. This is a continuous fermenter. See on this row, there are near about 10 fermenters are there, and these are the near about 4 lines are there. So we are taking with the help of the geotech photo. So this is a date as well as the satellite map is there. Okay. Now we are talking about the fed batch. What exactly that a fed batch one? We are very much eager about the different types of the fermentation. We know very well, either there will be the batch fermentation or either there will be the continuous fermentation. But it has not been possible every time go with such type of the mechanisms. Now this is the in between. Either neither be a batch fermentation, neither it may be the continuous fermentation. In this conventional system or the batch process, just describe all of the substrates which is get added at the beginning of the fermenters. An enhancement of the closed batch process is known as the fed batch of fermentation. Now in this fed batch fermentation, what exactly happens? Once there will be the batch fermentation, we never been added anything. Whenever there will be the continuous fermentations, you have to add the continuously, that will be the fermentation medium in the form of the carbon, nitrogen, minerals, and all these things. You have to properly design the fermentation medium, and then your fermenter will be at the zoom time. Now in this fed batch process, the substrate it can get added in the increments in the installments as the fermentation get a progress one. In the first time we are added the 5%, second time we are added the 10%, third time we are added the 15%. This is known as the installment. In the increasing of volume, we have to added the substrate. That's why it is known as the fed batch of fermentation one. Okay. Now, this fed batch fermentation is a modification basically of the a very simplest fermentation process. We call them as a batch fermentation. In this type of the fermentation, your substrate which is get added periodically in installments as the fermentation goes on, due to which whatever may be the substrate which is always at the optimum concentration. Why? Because most of the time what's happened when you are not taking the precaution about the concentration of the media in which we are added the source of carbon, nitrogen, minerals and whatever may be. For example, if your microorganism cannot tolerate more than 4% of the sugar, can such type of the microorganism they are tolerated and they are get growing as well as they are get multiplied? So never, because every system make as in the equilibrium in such a fashion so they can tolerate it up till to that only the concentration. If the concentration will be the more, at that time the same component they are responsible for the inhibition of the growth one. It is very simple on the bacterial growth curve. Whenever there will be the lacking of the media, at that moment the bacterial system undergo for the death phase. Whenever there will be the proper exact concentration of the all the essential component which is required for the life, at that moment only bacteria always been present in the logarithmic phase. They never been go into the what you call them as in the stationary phase. They never been go into the decline phase. Why? Because whatever may be the microbial mass is get produced there, at the same time all this mass properly utilizes the substrate one, and that's why they are always been present into the logarithmic phases there. That's why every time you have to maintain the optimum concentration of all the substrates one, and this is the essential, as some secondary metabolites are subjected to get a catabolic repressions by the high concentration. Best example that will be the sugar in the form of the glucose, as well as the another carbohydrates are there. I'm taking one of the example. There are the certain nitrogen compounds are there. Either that nitrogen compounds in the form of the protein molecules are there, either in the form of the amino acids are there, or either in the form of the inorganic compounds in the form of the ammonium sulfate is there, ammonium nitrate is there, etc. Whatever may be. So you have to always maintain, always thinking whenever there will be the designing of the good type of the fermenter, or you call them as on the bioreactor. And for this reason only, the critical elements which are get present in your nutrient medium, they are added in a low amount in the beginning whenever there will be the start the fermentation process, and this substrate later on continue to be get added in a small dose, just like today's era is there. Okay, yes, rest of the student, listen carefully. Your microphones are switch on, otherwise I will remove from your class. Please take the note. Yes, Kalyani, I am removed from the class. Vaishnavi Khante, please.
Yes, sir.
Unmute your microphones. It is a bad manner whenever the lectures are going on. And that's the reason is there. Why we are increasing the concentration. This is the best mechanism one. So in a small concentration, you have to increases this one, the concentration, so that the microorganism should particularly adopt that concentration levels one in a small doses during the production phase, because we are interested in the product. Okay, and this method is generally employed for the production of those particularly the substances one, the first time, that is the penicillin. Yashodhira, in 1973, the person introducing this term for the first time for feeding the substrate to the medium, as the nutrients most of the time it can get exhausted. So to maintain the nutrient inside the bioreactor at the optimum level, that is the reason why we are adding in an installment. This is the one reason, because most of the time your medium is get exhausted because of the microbial mass production is there. Large number of the microorganism cells, they are get present inside the bioreactor. Okay. Now the fed batch, a fermentation may be of three types, and these are the variable volume of fed batch, a culture. Second one, the fixed volume of fed batch culture. And the third one, the cyclic fed batch, a culture. Now, what is the difference in all these? The variable volume, the same medium is added, resulting as an the increase in the volume. For example, 10 ml, 20 ml, 50 ml, 100 ml, 200 ml of the same medium. That is a nutrient broth is there, whose composition you know very well. It containing the peptone, it containing the beef extract, it containing the yeast extract, it containing the NaCl, water, pH, etc. But you have to add it in the increasing volume. Now the fixed volume, a very concentrated solution, which is acting as an the rate limiting substances, which is get added in a very small amount, resulting in a significant increase in the volume of the media. That is a fixed volume. So 10 ml, 10 ml, 10 ml, 10 ml, or 25 ml, 25 ml, 25 ml, 25 ml. Okay, but why? Because of concentration of this solution, which is acting as an the rate limiting. We'll see later on what is exactly the rate limiting steps are there. The cyclic fed batch cultures, as it is not been possible to measure each and every the substrate concentration by following a direct method during the fermentation process, which is very essential as well as necessary for the better controlling the feeding process. Either it may be the indirect method are get employed, or certain that will be the direct type of the method which is get employed. One of the best example with the cyclic fed batch fermentation process, the production of the organic acid. I am talking about the acetic acid is there. Okay. Either it may be the citric acid is there. The pH value may be used to determine the rate of the glucose utilization. How this particularly the acid they are get produced from the glucose, that will be the what will be the rate of the glucose utilization from the media. That's why it is known as the rate limiting steps are there. So it is a very essential. This is one of the example. That's why that will be the cyclic fed batch culture method is there. Then what is an advantages of this such type of the fermenters? Production of the high cell densities, or you call them as an the mass, due to this extension of the working time, particularly the growth associated product. So most of the time, whenever there will be the large amount of the growth, there will be the large amount of the product is there. And the second advantage, control the condition in such a fashion, you have to provision of the substrate during the fermentation, particularly in respect to the concentration of the specific substrate. I'm talking about the carbon source. I'm talking about the nitrogen source. I'm talking about the minerals. Either it may be the organic minerals are there, or either it may be the inorganic minerals. So we have to check all these, particularly the phenomena. And this is one of the advantage mechanism. Control over the production or by the products of the catabolic repressions, effect due to the limited provision of the substrate solely required for the product formation, not necessary every time all entire carbon which is get exhausted, not necessary. What will be the mode of operation? Can overcome as well as the control deviations in the organism to show the growth pattern as found in the batch fermentation. Allow the replacement of the water loss either by the evaporation process. Alternative mode of the operation of such type of the fermentation which deals with the toxic substances or the low solubility compounds. So there are the chances one. So there may be the production of the toxic substances are there. Increasing of the antibiotic marker plasmids, stability. We are using the certain recombinant DNA bacteria or the microorganism which can produce the correspond antibiotics during the time span of the fermentation. Either it may be the 7 days, it may be the 14 days of the fermentation. Within that time, there is an 100% increase in the antibiotic marker plasmids. No addition, the special species of the equipment, it is required as compared to with the batch fermentation. So there is no necessity of the agitator, motor is there, impellers are there. Okay. There is no particularly what you call them as the baffles one. Okay. So these are known as the certain, the additional, the requirement which is required for the batch fermentation. Why? In that particularly the case, there is not the proper mixing of the entire media as well as the microorganism. While in this fed batch fermentation, there is the high quality as well as the sense one, there is the proper mixing. It is one of the effective method for the production of the certain types of the chemicals. I'm taking one of this broader example, that is an antibiotic, a penicillin one, which is get exhausted one during the process. You have to maintain the optimum level. Then once you are studying the advantages, naturally every system having its own disadvantages also. It is not possible to measure the concentration of the feeding substrate by following the direct method. These are the chromatography. We are taking the certain samples during the fermentation, and at the same time we are analyzes the concentration of the certain carbons one. Either that will be the glucose one, or either the nitrogen in the form of the amino acid. Go with the thin layer chromatography, or either it may be the paper chromatography. Run it. It's only show there is a presence of the carbon or not, but it do not indicate that what is on the exact concentration. This is one of the drawback. It required the precious analysis of the microorganism. It requires and understand its own physiology. With the productivity is an essential. You have to study the each and every the biochemical pathways, and that's why it required the precious analysis of the microorganism. And you know very well to understand its own physiology. It required the substantial amount of the operator, a skilled person, to set up the fermentation as well as the development of the process one. Without skilled person, never handling the fed batch type of the fermentation process. In case of the cyclic type of the fermentation or the batch culture, care should be taken whenever there will be the designing of the process that the toxin do not get accumulated. Okay, so this is very essential one, because the same thing is acting as an the inhibitory level, as well as the nutrients other than those incorporated into the feed medium, because limiting also, if many cycles run, the accumulation of non-producing or low producing variants may get a result one. So these are the certain disadvantages of the fed batch of fermentation. Okay, so in a coming lecture, we will study the aerobic type of the fermentation. So any problem? So student, any problem?
Sir. No, sir.